I think you can let me know, but I will be recording all of these sessions, and this is just to give a copy of this recording to the other faculty or the other teachers that are doing this program. All right, so let's go ahead and start. Let's go back to where we left off last time, and we're going to be talking about processing the FASTQ files, these files of gene expression, and we're going to try and look at the expression of genes based on analysis of these FASTQ files. So I hope everybody remembers from last time what FASTQ files are and kind of remembers some of the things that we covered, but even if not, I'll try to review some of that. So processing of next generation sequencing data in the context of transcriptomics really has two aspects. And sorry, Mia. Yeah, for now you can use the EDU site, but it would be much better if you went to the ScienceCoach site, because really we want to develop that material further, and we will be tracking activity on the ScienceCoach site. So I'll show that at the end. So processing next generation sequencing data is, in the context of transcriptomics, is going to be kind of two major aspects to it. One is we have to pre-process and map the data on the reference genome. And then once we map that data, we need to understand how to quantify the level of expression, right? So what is a gene again? A gene is a sequence that will be converted to a protein. And so when we are looking at transcriptomics, we're trying to study abundance of mRNA, messenger RNA, which is RNA that we can sequence, but essentially we're kind of anticipating that mRNA to become a protein, a functional protein. So first of all, to kind of review the process of sequencing, what is next generation sequencing? So next generation sequencing is when DNA, and in the case of RNA, it's converted to cDNA, so it's still going to be genomic data, but the sequence is sheared in small reads, short reads, and these fragments are prepared oftentimes as pair-end reads. So this is an example of a paired-end sequence, a library of paired-end reads. And then those are sequenced, and there are several intermediate steps, like, for example, PCR amplification, adapters, and things like that. And so in the end, when this information, this raw material, prepared material, is put inside the sequencer is able to read that signal by looking at different colors. And those colors are translated into letters. So as a result, we get this FASTQ file, and the FASTQ file actually contains sequence information as well as a quality of that individual letter. So as a result, you get a FASTQ file which has many, many different reads. Now, once you have those reads, one of the first things that you can do is align those reads onto the reference genome. So this is an example of a file like this with FASTQ aligned to some reference genome. And what you can see here is abundance of reads, right? So here you have a lot of reads aligning to this region. Aligning to this region, you can see that there is an abundance, and you can see also the types of reads. So the blue ones, blue and red, are going to be those paired end reads. So reads that go one direction or the other direction. And sometimes you will see these black ones, and the black ones are going to be the ones that don't have a matching pair. So this is just an idea of, okay, what are we looking at in terms of data? Now, in terms of preparing this data, and we went through all of this last time, really just to kind of reiterate what we're doing is we're trying to identify which genes, which are certain locations on the full DNA, on the full genome, which genes are transcribed into transcripts, into genes, but in mRNA, not on the genome, not on the DNA, but in mRNA format. And we need to understand in the context of the sequencing process how the preparation of the data plays an impact in our analysis of the data. When we build that pipeline, how does that actually impact our analysis steps? Because we want to ultimately know which genes are being transcribed and how much. So how many copies do we have of this gene that are converted into mRNA? This will give us some indication how much of that is being used or how functional that gene is in a certain set. Now we can then do a lot of things once we have that data. We can compare different patterns. We can identify different patterns associated with different conditions. We can do a lot of things. But first, we need to prepare that data set. So to prepare that data set, basically we need to have first three steps, right? We need to remove the adapters, and then we need to remove the PCR duplicates. After that, we have to map the reads without the amplification and without the adapters. We need to map it on either the genome, the full reference genome, but that's a computationally intensive task. And also, we know that a lot of regions on the genome are not going to be expressed. So another strategy is we can map this on the transcriptome, on just the expressed portion of the genome. And then we, of course, need to quantify the levels of expression. Now, does any of this make sense to you? And, did you have any questions from going through the materials online on the site? Does anyone have any questions about this so far? And I know it's a little bit, different when we are doing this online, but I do really want to try and maintain some level of interaction with all of you. So please give me some feedback so I know that, we're actually moving in the right direction. We're not losing anyone because some part of it is not clear in some way. Okay, we're good. Okay, thank you. And Mia, did you, is this all? I know that you've been doing some, I saw that you're a little bit more active there on the learning side. So all of this is clear so far, Mia? Okay, Ethan, thank you. Nicholas, thank you very much. Mia, everything is clear? All right, okay. Well, so now, why don't we try and do what we are doing here as a pipeline. Now, as you remember last time when we were during the workshop, we used a demo pipeline. So we'll first do that, but then I want to show you how to upload some data, where to find that data, and how to prepare a pipeline because it will require several additional steps. So last time during the workshop, what we did is we prepared this as a mapping on what, right? So I'm going to ask you some questions just to kind of see how clear everything is. But I will show you two different pipelines, and let's see what the differences are. So what is this pipeline? Who can explain what does this pipeline do? And by the way, if you want to, you can unmute yourself. You can turn on your video if you would like, but you don't have to. But I'm just saying, or you can put it in chat. That's also fine with me. But who can explain what does this pipeline do? Okay, so I'll do the first one just so it's clear to everyone what it is. Let's go back, right? So let's just take a look at the color coding. Blue is pre-processing, which is just eliminating some technical variation. Red is mapping, so we need to align our reads to the genome or the transcriptome. And green is quantification of expression. Now, how do we read this on this flowchart? On this flowchart, we can see the blue, right? The blue is pre-processing. And then we see the red. The red is mapping, right? Now the question is, what are we mapping to? Right here it says mapping on transcripts. So if it's transcripts and not mapping on genome, we know that this is mapping on the transcriptome, right? And then the green The green is quantification. So here we see expression levels. So now we are quantifying the expression of those transcripts. Okay, so that would be the right answer. Now let's take a look over here. What does this pipeline do? Who can tell me? Does anyone want to try?
Hi, can you hear me?
Yes.
Okay, so the pipeline is taking the first step, which is pre-processing, and then it's mapping on the genome and mapping on junction. And then it's going through isoform construction and GTF processing, mapping on transcripts. It's doing a lot. And then it's expression levels, and then that's it. Am I right?
Great. Yeah, perfect. So now we know how to read this pipeline, right? And very correctly, Mia has identified that what we are doing is an expanded pipeline that's actually taking several steps that seem to be all combined into a very concise pipeline before. So let's go through some of these steps just so that we're all clear about what they actually do. So nothing changed in the pre-processing step. We're still removing the adapters and then removing the PCR amplification. Then we are mapping combined on genome, exon detection, and mapping on junction. Then we are constructing those isoforms. Again, what are those isoforms? Isoforms are the actual transcripts of genes and not the genes themselves, because you have a sequence, but that sequence is transcribed in several different ways, in different combinations of exons. So that's what we're doing here, is we're constructing those isoforms. Then we have to combine all that information into the GTF file, which is where all the beginning and the end of each gene is written down. And now we can do the same thing. We can map on transcript. Now, why are we mapping on transcripts? Because we want to quantify the level of expression. Now let's prepare this pipeline. So how do we prepare this pipeline? We go here, okay? And the first thing that we'll do is we'll do this as a demo. Now you do have to be logged in. And I believe Joshua prepared the accounts also for Ethan and for Nicholas. So at this point, I think everybody should have access, apart from maybe Hannah and Trevante, that were not able to open the site. But if you have it on the desktop, right, this is something that everybody could follow. So right here under transcriptomics RNA-Seq, we can click on the drop down. And first of all, we can go to the cell line project. And we can click on start. Here we can see all of these steps that we are trying to prepare. And first, let's prepare a simple pipeline. So we'll go to Trim-O-Matic, which, as you remember, we need to remove some adapters. So you can see that the blue ones are adapters, and there's some, there's the valid sequence and then a trimmed sequence. So we're really looking at cleaning up those read sequences. And then we are reducing this PCR amplification, this step right here. Then we can map on the transcriptome, and you can see how those reads align to the reference genome. Okay, and you can see that this is the forward read, the backward read, right? Okay, and then we prepare an RSEM expression table, which will look like this, where you have gene ID, and then you have level of expression. Click on OK, and then on End. Okay, now let's take a look at the output data. Right here, you should have output data. So you just click on it, it downloads, it's a pretty big file, but let's take a look at what's inside. Okay, now, does everyone get this output? Did everyone find this file? Type yes if you found this file. Type no if you cannot find this file. All right, Tiska, thank you very much. Anyone else? Did anyone else find this file? Okay, let's go through it again. So, Nicholas, did you get access to the platform? Did you receive an email? Okay, so okay, Nicholas did. Ethan did not figure out how to sign in. So, Ethan, let's see. Did you? So, if you go here, you should be able to log out. So let's see. You should be able to click on login, and then you were sent. Oh, okay. Okay, so Ethan found. Okay. All right. So once you log in, you should be able to. Let's see. No, I forgot how to log in. Okay. You should be able to click on this drop down, go to Demo RNA-Seq, click on Start, Trim-O-Matic, PCR Clean. Then we did Bowtie 2T, RSM expression table, and then End. And once you click on OK, you will get this download pipeline output files. You have to click there, and then it opens up and you have this gene expression or cell lines expression data. So Nicholas, does that, did it work this time for you? Okay, great. All right, so we downloaded this data. Ethan, were you able to log in and get the data? And did we lose Mia? Okay. Okay, great. Okay, so now we open this data, right? Let's just open it up to look what's inside. And this is the gene ID, right? So the gene IDs, and this is the actual expression level of that gene. Okay, now what we want to do is we want to look at, for example, this is a name of a cell line, right? So this is the name of a sample. We want to say, okay, well, how is this gene expressed in these different samples? Because we have a lot of different samples. So what we can do in Excel or any kind of table manipulation software, we can compare, for example, just visually, right? We can see, okay, the expression levels go up and down based on which sample they are in. All right, so all of this we can read about in these courses. So let's take a look. Where is it in the courses? So this is the page for courses. And in Transcriptomics 1, you should be able to take this course, press it here. And now what we are trying to do, so here you can read a little bit about what we are doing, right? And how all of this works. And then you should be able to read about the pipeline preparation, but it's going to give you a file. So let's try and do that. So here, it tells you about the data set. What is the data set? Now, this data set is different from the data set we started looking at during the previous workshop. It actually is a data set from a human tumor that is being grown inside a patient-derived xenograft model, so a mouse model that inside has a growing tumor from a human patient. And you can download the data set right here by just clicking on this gray box. And what it will be is actually a zip file. So you can see that it's a zip file of an SVL file. Okay, so let's take a look. You unzip this file, and you get this file that's called SVL. Now you can open this in a text editor, and you will see that actually inside it has a lot of different files. Now, what we want to do, you can look at the naming of each file, and you will see here several things. So first thing that you will notice is that each name is repeated twice, but then it has an _1 and _2. What that means is that this is a pair-end file. So the sequencing, the library preparation, was done with pair-end sequencing. Now, as you remember, pair-end sequencing, let's go back here. Paired-end sequencing is when you have two copies of each read. One is a forward direction, one is a negative direction, or forward and backward directions. By combining those together, as you can see right here, we get a more accurate reading of each letter. So because we see that there's two files for each sample, we know that this is paired end. The other thing that you will notice is that there are two types of samples. One is ER. As you probably know, ER is a receptor. You can be estrogen receptor positive or estrogen receptor negative in the case of many epithelial cancer conditions. And TN actually stands for triple negative. So triple negative breast cancer. We talked a little bit about breast cancer last time, so you probably remember. Now you can see that there's a lot of files, but these are just links to files. They're not the actual files. So you should now be able to go back to the platform and instead of doing a demo, create an actual pipeline. So how do you do that? You go to the areas of analysis, and please, if anyone is lost and kind of following these steps, just stop me and I'll be happy to review again. So we go back here, and instead of clicking on this drop down, we just click on the name itself. And now we have to make some settings to be able to prepare a pipeline, right? Because if I just click on start, it will tell me, please upload files. Okay, great. Mia, great to have you back, and sorry about your computer. All right, so now what are these settings? To understand what these settings are, you hover over this eye, and you should be able to click on it, and it will just give you some pop-ups. But basically, I'll explain to you what this is. First setting is what type of genome, what type of reference genome are you going to get? So you can have three types of reference genomes. One is a model genome, a model organism. Here are some model organisms that are already pre-uploaded on the genome, on the platform. Then you have single or pair end reads. So single or paired end reads, what type of reads do we have here in our file? Who knows? Do we have single end or paired end? Paired end, okay, thank you very much. So we just change it to PE. And who knows, do we have FASTQ files or FASTA files, SAM, BAM? What should we use here? FASTQ. All right, thank you, Ethan. FASTQ files, that's correct. Now, which organism do you think this is? A little bit of a more difficult question, but look at your options and let's think together, what kind of an organism would this be? Human plus mouse, right. So right here we have human mouse concatenated. Now, does anyone know what concatenated means? I'll take it as a no. Concatenated means that you have exactly added together in the sense of the genome. What it means is you take a sequence of the human gene and then you place the sequence of the mouse gene. Human gene, mouse gene, human gene, mouse gene. So it's added together, but not like all of the human genome and then all of the mouse genome. It's one after the other. So that's the human mouse concatenated genome. And now you can see that I can proceed to step 2. So I can do file upload. Now, when we upload files, there are several options, and this is going to be very relevant for your project. So we have several options. You can upload files like from my computer. I can just click on add files and find some FASTQ file. But of course, that's going to be a very big file. I don't want to do that right now. I can add files from a link. So for example, if I have a link, an HTTP link or an FTP link, I can just paste this right here. Or I can also use an SVL link. SVL link is a link to a file that is already on the server that we're using. And that's exactly what we have in this file that we downloaded. Links to files that are already on the computer. So, but instead of doing them one by one, which we have a lot of them, you can do bulk upload from an SVL file. So what we need to do is we can click on this button and select the SVL file that we downloaded from the educational lesson. Okay, if you see something like this that creates 2 of them at the same time, you just do cancel and try again, because it should only be 1, just like this. Okay, so make sure it's only 1 that you see here. And now you can go to group selection. Okay, did everybody get to this point? Please just put in yes, but I'll continue. Now, as you remember, we have 2 types of samples here. We have ER, right, and triple negative. Now, we could just create a table of expression, or we could create a comparison between them. If we wanted to create a comparison, we would create 2 groups just by clicking here, and then drag the relevant files in the right box. But right now, we're not going to do that. So let's just press on cancel, no groups. Now, groups have been added, but we don't need groups, so we are not going to use differential expression. Now, let's recreate our pipeline, the one that we had from before. Okay, did everybody get here? Yes. Okay, great. So just for the purpose of going through this again, I will quickly do this. Human mouse concatenated, file upload, URL bulk upload, upload the SVL. It uploads 1 time, group selection, cancel no groups, start. All right, and now I have a lot of options. Now I have to make decisions based on these options and what I have uploaded. So first of all, I know that I need to remove the adapters from Trim-O-Matic, right? So I need to click on yes. Where do I go next? Who can tell me? PCR clean, all right, yes, let's do PCR clean. And then after PCR clean, what I will do for the purpose of saving us some time is I will go to bowtie 2. And then I'll go to RSEM expression table, and here you will see that I have a few options as well. So what are these options? There's fragment per kilobase per million, expected count, and TPM. We'll learn about this more later, but right now let's just select that and click on end. Now you see that I need to save my pipeline. So I'm going to write here PDX models transcriptome, right, because I'm mapping on transcriptome, and then gene expression RSEM. Click on save, run pipeline. Now you should see that it has started. And when you go back to my pipelines, You should see right here that it is processing. So I can go back inside and I can see that it is processing. Did everybody get to this point? All right, excellent. Now let's see how these two things connect. So if you close this lesson, which by the way, you can go through this lesson and kind of understand in greater detail what's going on, all of us should be added to this pilot program right here. And the pilot program is a group that we have where you can see all of the activity that is happening. So for example, Ethan passed the course Transcriptomics 1, etc. Now we're going to be working on this to make sure that all the activity is here, but basically what we should be able to see are the members in this group and all of your activity. So what we want to do is we want to be able to, okay, so now, yeah, we see everybody is here. And what we want to be able to track is everybody's progress in terms of the curriculum, as well as in terms of making these pipelines. So whenever we do demo, that doesn't count. You don't get any points for it. But when you run an educational pipeline like this, that counts. So as you progress in your learning, you'll have different types of pipelines to create. For example, now you can create two pipelines, right? One like this and one like the expanded one, and then you'll get, points for both of them. So that's a little bit on processing this next generation sequencing data. Let's talk a little bit more about quantifying. What are we quantifying here? When you finish that pipeline, you will get this kind of an output, FPKM. So you can see that we're looking at FPKM. Now, what is FPKM? FPKM is fragments per kilobase per million of mapped reads, which means that we are going to take the number of all the reads that are mapped to a specific gene, and we will divide them by the total number of mapped reads in millions, multiplied by gene length in kilobases. Meaning that if your gene is longer, you will get, statistically speaking, you should get more reads aligned to that gene, right? It's like having a big bucket and a small bucket. If I just keep throwing reads by random, by chance, I will get more reads in a big bucket than a small bucket, right? So in the same way, we can account for that difference because of the gene length. So what are the two factors? Imagine I have rain, just shower, let's say, and I have gene lengths, right? Big bucket, small bucket. But another aspect of it is how much rain do I have? How much water am I trying to pour here, right? The more water I have, the more expression I will be able to quantify, right? So if I want to take two samples, one had less reads, another one had more reads, I have to account for the total number of reads I have in each one, and that will help me compare the two samples. So in the same way, we can use another type of a transformation just called TPM, transcripts per million. So transcripts per million means I'm just taking a million as a universal metric that will help me make those two samples more comparable. And then, of course, you can just use raw counts, which means I ignore the length of a gene, I ignore the total number of reads that we have, and I'm just looking at how many I can count. But then, of course, I need to normalize some of that data. Okay, so as a result, you will get something like this. You can see here ENSG, again, is the gene ID. And these now, what are these? How is this different from the cell line data that we were looking at? Who can tell me? Does anyone remember what we got from the demo pipeline, and how is this different from what we got here? Anyone? Okay, well, if you don't remember, okay, are they levels of expression of certain isoforms? Well, yeah, so these are genes, but now instead of, as you remember, the first, so let's go back to Excel really quickly, and let's take a look. In our cell lines expression data, these were cell line names, right? And the cell lines, we just had names of individual cell lines. But here, when we look at this table, we actually see ER, right, and TN. So now we see samples, the names of the samples changed, and we actually know that there are a couple of groups here. Now, you also see a lot of zeros, right? So you have a lot of zeros. For example, this is a line. You can see that for this gene, there's absolutely no expression, right? So how is that? How is that that we have some genes that have no expression at all, some genes that have some expression, some genes have 259 or 1939, and some have 1.38, right? How does that work? So let's try to understand this a little bit better. This is the GTF file. So the GTF file, you can see that it has the gene ID. Now this is a common gene name, not like we have in Ensemble ID in our table, but this is a common gene name. But it actually is broken down by exon. So you can see that this is an exon, the beginning position, the end position, and then here you have gene ID and the number of exon within that gene. So what is happening here is that we have positions, the beginning and the end of a certain fragment, and then we calculate the level of expression. Now, since we have a position, that becomes an entry in our table. Even if there's zero reads aligned to that position, it's still going to show up in the table. But we also have some that are going to have a lot more and less, and so we record all of the genes in our GTF file are going to be present. So since they're all present, what you should expect to see are what two types of genes? Now, this is a complicated question, but think about it, how we prepared the pipeline. What two types of genes are you going to expect to see in your expression table at the end when your pipeline finishes? Who knows? Think about this GTF file, right? So the GTF file are genes from the genome, right? So what two types of genes do you expect to see in your expression table? Exactly, human and mouse genes. Thank you very much, Ethan. And you can tell that Ethan worked on his course. So we can see that we expect two types of genes, a human gene and a mouse gene, at least those two types, right? All right, so then let's take a look here. How do we find those genes, those human and mouse genes? Well, since we're using ensemble IDs, it's very simple. You have ENSG, which stands for human, and ENS mus G, which is for mus musculus, the scientific name for the mouse. And this is going to be ENS mus G. So those are going to be the two types of genes. And you can see here two examples. Once you finish your pipeline, you will see this is a human gene, this is a mouse gene, and you can see the expression. Okay, now let's think about it for a second. What are we actually trying to do in the context of this project? And this is going to be now something that is going to turn into homework assignment, okay? It's not going to be graded, but it's something to let you practice on your own. Let's go back to our lesson. Okay, so in the lesson, you will have an explanation of where this data came from. And what's very important when we analyze this data is that we don't detach the data from what we are trying to study. Now, Ethan probably already knows this because he went through some of these, but let's, for all of us to kind of review, let's take a look at the data. So we'll take a look at the paper where the data is being described. Okay, so you can see here that the data set is an educational data set. In other words, we modified the data set. It's not the original data set, but it's based on the real data from this publication. So you can click here. So let's open it up. And you can see that this is a publication on PubMed. So you can get your full text right here. Okay. And let's try and read quickly through this abstract to understand what were they trying to do. The tumor microenvironment is emerging as a key regulator of cancer growth and progression. However, the exact mechanisms of interaction with the tumor are poorly understood. Whilst the majority of genomic profiling efforts thus far have been focused on the tumor, here we investigate RNA-Seq as a hypothesis-free tool to generate independent tumor and stromal biomarkers and explore tumor stroma interactions by exploiting the human murine compartment specificity of patient-derived xenografts. Okay, let's convert this to English. So tumor microenvironment, I think, is pretty straightforward, right? You have a tumor that is growing, but it is surrounded by an environment. Now, if you think about the PDX models, these patient-derived xenograft models, where is the tumor coming from? Where did they get the tumor? Who knows? From the patient, right? So was the patient a human or a mouse? A human, exactly. And they implanted this into a mouse, right? So you have the tumor and you have the environment of the tumor. The tumor is from the human and the environment is from the mouse. So now you can look at how to separate what happens inside the tumor from what happens in the microenvironment. So now that we understand this, we are talking about interaction, right? So what is an interaction? What kind of interaction could there be between the tumor and the microenvironment? Well, the tumor needs resources to grow, right? So it needs blood and it needs food and it needs to be not attacked by the immune system. All of those aspects, the immune system, the blood, the food that comes to the tumor comes from the microenvironment. So how could it be? Tumor is obviously very dangerous to the body, it's harmful to the body. How can it be that the body doesn't stop the tumor from growing? Who knows? Who wants to guess? How is it that the tumor is not stopped by the body from growing? The body sees the tumor as a normal part of the body. Well, that's correct, but the reason is because the tumor and the body actually communicate, right? So since this is a tumor from a human, it's very obvious that the mouse is going to see this as something foreign, right? So the tumor actually has a way of communicating with the cells around it to stop this kind of attack that should naturally occur, right? So it should stop the attack of the immune system, which is done by the means of communication. It should then also tell the body, because it's growing much faster than the surrounding tissue, it should tell the body, give me oxygen, give me food, give me this, give me building materials, right? So it's starting to communicate and create communication pathways that tell the body to recruit the buildup of blood vessels, the buildup of the surrounding tissue that can bring it all kinds of resources. So that's what's going on. And this kind of interaction is not understood very well. I mean, there's a lot of questions about why does this happen? How does this happen? Why does it not, stop? Because it's obviously harmful. So that's what we're trying to understand. And a lot of times when we think of cancer, actually people are much more focused on the tumor itself, which is what it says right here. But we want to use RNA-Seq as a hypothesis-free tool to generate independent tumor and stromal biomarkers. So just right here, we have a few interesting words. What is hypothesis-free tool? What do you think does that mean? And anybody's welcome to answer, of course, encouraging everybody else to contribute. Obviously Ethan went through some of this already, but anyone else, Mia, Nathan, or Nicholas, sorry, Tiska, anybody else want to try and answer this question? What is a hypothesis-free tool? Means their research doesn't have a guesstimate or hypothesis in their study yet. They're just searching. Approximately, yes. So it means that hypothesis is an idea, framework that we can prove or disprove, right? So for example, I can say, well, it's this one gene that is responsible for something, right? And then I can easily prove or disprove. Find evidence for this being there or find evidence for this not being there. But hypothesis-free, what makes RNA-Seq a hypothesis-free tool is because we are collecting data about all of the genes that are being expressed. We're not just looking for those 5 genes. As you remember in our previous workshop, we looked at PAM50, or we looked at Oncotype DX, right? So we looked at these panels which were carefully curated, selected to tell us basically an answer to a question. Do you have a cancer of this type? Do you have a cancer of that type? Do you have a cancer, right? What is your survival curve, right? So now the RNA-Seq is different from those panels. It's a hypothesis-free tool. It generates data, and now we can search the data for biomarkers, right? So biomarkers would be those types of genes or pathways that have an indication about a process, about something that is happening. And what we want to explore is the tumor-stroma interaction by exploiting the human murine compartment specificity of patient-derived xenograft. So human murine compartment specificity basically means what we already said. We have 2 genomes. We can map the reads on one and the other, and now we can look at just the mouse genes, just the human genes. That's all that means. Okay, well, I encourage everybody to take a look at this publication, try to understand it for next time. Next time, we'll go a little bit deeper into what are these different types of samples that we're dealing with. We're not going to take all of the samples. We're just going to take the ER positive and the triple negative. So try to understand what are these and why do we need to know that one receptor is expressed or not expressed. And also try to understand what this data looks like. So I think by next time, everybody's pipeline should finish, right? So you can always go back to my pipelines and you will see how your processing percentages here are going to change, okay? Now, the next thing that we want to do is we want to start understanding this hypothesis-free tool for RNA-Seq, and we want to understand how to use different methods of analysis to be able to compare different conditions, understand what's in our data, and then be able to set up a comparison. So that's what we wanted to cover today. I encourage everybody, now that everybody can log in, to try and complete this first course, Transcriptomics 1. And next time, we'll focus on Transcriptomics 2. So you're also encouraged, if you want to try Transcriptomics 2 and play around with this data set, we're going to build on what we've learned so far. Now, does anyone have any more questions? Yes, go ahead, Mia. And you can go ahead and unmute yourself if you want to just speak. You don't have to type.
Hi.
Hey.
Okay. So I want to like experiment more with the pipeline and everything. So I just want to know. I'm thinking I'm gonna find more data sets on the internet and stuff like that. So what are the parameters of the data sets I can input into the pipeline? Is there a certain data type it always has to be, and that type of thing?
Sure. So what we're going to do is prepare you to do this in a meaningful way. And so right now what we want to do is first focus on just RNA-Seq type data. And we want to learn about processing because processing will give you an idea of what is the signal that you're looking at. Once we establish how to process data, which is always going to start from a FASTQ file, but FASTQ files come in a variety of formats. They could be compressed, for example, SRA. They could be already mapped. That could be BAM or SAM. Or they could be just FASTA file without the quality. So if you go into the transcriptomics, right here is a drop down that shows you what kinds of formats you can use in this specific pipeline. Now, everybody has an educational account, which means that you can only work with SVL files. You cannot upload anything bigger than 50 megabytes. Typical FASTQ file is going to be around a gigabyte or maybe even more. So you cannot like find a FASTQ file and just upload it. But what you can do is you can play around with this until you understand how it works. And once you gain enough points, then you will be able to do your own project. And I'll show you how to download a file from NCBI that will allow you to actually run a pipeline on your own data. But it will take a few meetings more to kind of get to that level. In the meantime, if you do have a gene expression file, which by the way, if you go to Transcriptomics 2 or even complete Transcriptomics 1, you will be able to download a gene expression data, process data, which will be in a TXT, a tab delimited format. And then you have a number of other data mining and annotation techniques here that will let you look at a statistical analysis or a machine learning algorithm or annotation of that data set using pathway analysis, for example. Does that make sense?
Yes, makes perfect. Oh, yes, it makes perfect sense. Thank you so much.
All right, great. Anyone else? All right, well, no questions. All right, well, great. Thank you, Ethan, and thank you, Mia. If anybody else wants to ask any questions, I can stick around for a few more minutes. If not, then I suggest that we meet again. So we have set up, let me just pull up our next meeting, and we will send an email invitation again for the next meeting. So let me just pull it up real quick, and I'll tell you the date. Okay, so for our next meeting, ScienceCoach. Okay, here I found it. Our next is going to be on October 30th, statistical analysis. So for statistical analysis, we will start talking about, we'll talk about p-values, we'll talk about how to do differential gene expression, we'll talk about how to detect more complex patterns in our data, in the data that you will generate here. So what you can do is you can go ahead and do Transcriptomics 2, and you can run a couple more pipelines to get familiar with some of the other things. So I'll see you, it's going to be October 30th. Let's just take a look at the calendar. I believe it's going to be a Wednesday again. And I will also introduce somebody else that will help in teaching. So next time we will go through that. So Wednesday on the 30th, we will have our next meeting, again from 4:30 to 5:30. And I hope to see all of you there. Okay, so thank you again for joining. I'll stop the recording.